I give you a point if you turn it in, is the way it works. So I shouldn't say that, should I? Okay. Okay, where are we? Okay. Let's go through the other forms of selection, and these are different kinds of selection. This is what we call overdominance or heterozygote advantage, and that's pretty simple to understand, right? Which genotype has the highest relative fitness out of these three? The heterozygote, and two homozygotes have a fitness reduced, relative fitness that's below 1. So this is what we call overdominance or heterozygote advantage. There are very few instances of this. Sickle cell is one of them. There's some suggestion that cystic fibrosis may have some sort of gene that's associated with heterozygote advantage, but it's a fairly rare phenomenon. And we seem to spend an inordinate amount of time talking about it, and I don't understand why. But anyway, so it's important. But one of the sickle cell gene is, and malaria, interaction with malaria is one of the most interesting cases. But here are the key things. No matter where you start, whether, again, it's the frequency of the A1 gene, no matter where you start, what's the outcome? Is there an elimination of the A1 gene? If you look at this graph over 140 generations, nope. So what you get is a stable equilibrium. And the idea here is that it goes to some value where it becomes, in this particular case, becomes stabilized after about 60 generations, no matter where you start. So if you start at a low frequency of the A1 allele, it moves towards that equilibrium value, and that's probably about 0.35 maybe. If you start above that equilibrium value, it declines down to it. So this is what we call a stable equilibrium. And so this can be an interesting case where what happens in overdominance or heterozygote, A1 eventually reaches an intermediate frequency that does not depend on the initial frequencies in a population, right? Whether it's near 0 or almost near fixation, so long as both alleles are present in the population. Okay, so turns out this is a fairly rare phenomenon. Now, how is this different from dominant selection, intermediate dominance, and the recessive model? What's different about this one in terms of the outcome? Well, the dominant takes, not really, the heterozygote has a fitness advantage, but what happens to the frequency of the alleles? Does it go to do you get the same outcome in this case as opposed to the previous three, or what's different? One, the previous three you get fixation or loss of alleles, right? Here you maintain both alleles in the population. Okay, so that's kind of one of the big differences. You maintain both alleles under heterozygote advantage. And we're going to show you another example called frequency-dependent selection that has the exact same outcome in that it maintains both alleles or maintains what's called a polymorphism. It maintains both alleles. So one of the classic examples of this in terms of a stable equilibrium is the sickle cell gene that is associated with, obviously it's a huge health issue in Africa, African American populations, and is a really, really debilitating disease with huge health consequences, and it's something that a lot of people are very interested in. This is probably all moot now, and people think they're going to somehow use the CRISPR technology to actually eliminate the sickle cell gene so that's what Bill Gates is spending billions of dollars on. So anyway, we'll see where that all ends up, but Bill Gates it's one of his biggest health initiatives right now, and whether he'll use this new gene editing technique, we'll see. Okay, so anyway, so what's really interesting about this is this is a very nasty allele or variant. It's called the sickle cell gene. And we know the consequences are huge health risk, coagulation, and pretty high mortality. Okay, it's a very debilitating disease. It's most debilitating when you have two copies of the sickle cell gene. That's when you get the anemia and all the really bad consequences, okay? So this is called the sickle homozygote, right? And then there's a homozygote where you have the normal copies and not the mutated copies to the sickle. So you have two homozygotes, and then you have the heterozygote. The idea here is, and we're not going to go into all the details, is that this sickle cell gene in African populations, the sickle cell allele reaches pretty high frequencies. Around anywhere, it depends on where you are, West Africa, Central, South Africa. It depends. There's a lot of variation among African populations in terms of the frequency of the sickle cell gene, but it can range anywhere from 10 to 25%, just that variant. And you'd think, your first thought is, right, shouldn't that be eliminated through natural selection or things like that? Well, it turns out what happens is under heterozygote advantage, it turns out those individuals that have a copy of the non-sickle gene and the sickle gene have actually do you know the story? Why do they have an advantage? Yeah. So there's a higher resistance to malaria in that are heterozygous. Okay. So here you have to think about disease. Here you have malaria caused by, obviously disease, and then you have these other consequences from this sickle cell gene. So in these two different contexts, what you have is the heterozygote has an advantage because it's more resistant to malaria. Okay. But again, you have a problem, right, in that, those individuals, when they mate with other heterozygotes or, those sorts of things, can produce a lot of individuals that are homozygous for sickle cell that have really high mortality. And so, again, this all varies a lot, and I've just put these up there. It depends on the population. But here, what's envisioned is the relative fitnesses of these three genotypes. And again, it's not this simple. There are a lot of environmental influences on outcomes, and this is not, what we're presenting is a very simple approach, is that the idea here is that individuals that have two copies of the sickle cell gene have a selection coefficient of 0.7. That means they have a relative fitness of 0.3, and that's attributable to mortality and all these sorts of things. Some individuals do survive some period of time. It depends on health care and medical care, and that varies a lot depending on where you are. But the non-sickle cell homozygotes, right, do they have a fitness of 1? No, they have a reduced fitness of 0.8. So their selection coefficient is 0.2. And that's presumably because they're more prone to malaria. They're less resistant to malaria. And so they suffer from the consequences of malaria. Whereas under this model, which genotype has the relative fitness of 1? The heterozygote. Okay? So you can actually determine this. And there are ways to do this. You can actually determine what should be the equilibrium value of the sickle cell gene, okay? And it turns out it's the selection coefficient against, and you're going to say, why is that? Well, don't worry about it. It's S1 divided by the sum of the two will give you the equilibrium frequency of the sickle cell gene under these kinds of fitnesses. And so in that previous model, it went to about 0.3. The idea here is that the equilibrium frequency in sickle cell genes, and it varies among populations, but you can see the presence of the sickle cell gene is pretty tightly associated with where malaria is endemic, right, across a lot of different regions. So there's a strong association between that. The idea here is the sickle cell gene should reach an equilibrium of about 0.22. And what's the equilibrium frequency of the non-sickle cell gene? 0.78, okay? So so the sickle cell gene can reach an equilibrium as high as 20% or even higher in some African human populations. So it's something a lot of people are interested in, trying to provide some much better medical care approaches and health to deal with this debilitating disease. And it's a problem in African American populations. Well, not really. Those selection coefficients are based on mortality rates, and it depends on where you are. But when you go in the absence of modern health care, right, and probably the historical patterns, right, before Western medicine arrived in Africa, most of the deaths occurred before reproduction from malaria. So that's kind of how those models were done. And it's changed a lot. Okay. Does that answer your question? Okay. Yeah, forget that. Don't worry about underdominance. Okay. So ignore that. It's pretty straightforward, but it's not a common model at all. Okay. Or it's not a common outcome in nature. This is what I think one of the more interesting kinds of what we call frequency-dependent selection. And what I'm going to focus on is what's called negative frequency-dependent selection, and let me just define what that means. What that means is there are alternate phenotypes and or genotypes in a population. Their fitness is not fixed. And negative frequency-dependent selection means that those individuals, genotypes or phenotypes that are rarer or less common are favored by selection. That is, they have a higher relative fitness. And then that switches as they become common. It goes back to the other way, okay? So it turns out, and we'll be spending a lot of time on this when we get to the second part of the course, this is believed to be a very important mechanism of selection in interactions between hosts and parasites. And we'll be talking a lot about this and how it maintains diversity, especially in parasites. And we'll be talking about this. This is becoming to be recognized as one of the most common forms of selection in nature, besides what we call directional selection, all the other things we've talked about. Okay, so let's kind of just go through this, and then I'm going to show you an example of it. So it's pretty simple. What we're imagining, fairly simple. Let's look along here. Here's fitness. Here's the frequency of one allele. Let's just say we have, what do we have? P1 and P2, let's say. So again, you have 3 genotypes, 2 alleles, P1 and P2. And you can make these curves anyway. Here, the red curve represents the fitness of the P2 phenotype as a function of the frequency of P1. The blue represents the fitness of the P1 phenotype as the frequency of P1. So let's kind of look at this. So here's, is that right? Yeah. Yeah. Okay. What? I'm confused now. Okay, right. So here's what's happening here. Here you got a case, here's the fitness of the P1 phenotype, right? When it's relatively rare, right, in a population, down in here, this is the frequency of the P1. What's its relative fitness compared to the P2? Is it higher or lower? It's higher, right? So in this particular case, when this thing is rarer in a population, right, that means the frequency of P1, this is what got me confused as I was reading this. When you look at this, this is the case when the P1 is rare in a population. Let's say it's around 0.1. And the frequency of the P2 in this case would be about P2, okay? Would be around 0.9. What you can see is up here is that which one at that point at the frequency of 0.1, which one has the highest relative fitness? The P1, okay? The P1, let's just say the P1 individual, okay? Whereas the P2 has a reduced fitness. What happens as you go out to a higher frequency now in a population of the P1? Let's say you're out at 0.9. Which of these 2 phenotypes has the highest relative fitness? The P2, okay? So what all these curves are demonstrating is that the phenotype that's rarer in a population has higher fitness, okay? So in this case, the P1s have a higher fitness. Out here, when it's P2 is much rarer, it has a higher fitness. So the idea here is this is an example where the individuals that are rarer, the genotypes that are rarer, have the highest fitness. And eventually what happens is, you don't have to worry too much about the mathematical part, but basically when A1, for example, the A1 allele starts at a high fitness, frequency of P is common, it basically starts to decline over time. And actually what it does is it reaches an equilibrium. This doesn't show, this is what's really confusing about this graph, is that basically what you get is A1 eventually reaches an intermediate frequency and reaches an equilibrium where both things are maintained in the population. That's the key thing I want you to take home from this, is that basically what happens is these rare genotypes, right, increase in frequency. The common ones decrease in frequency, but they eventually reach an equilibrium where both alleles can be maintained or both phenotypes can be maintained in the population. So this is kind of like overdominance. You're not losing one allele or fixing another allele. You're actually maintaining both phenotypes and genetic diversity in the population. Let me show you, I think was the best documented example of this and kind of put something real to it. These are fish from Lake Tanganyika in East Africa. In East Africa, there are all these rift lakes, these huge rift lakes that probably originated in the last, they think, anywhere from 10 to 100,000 years ago, maybe 400,000 years ago. They now have, in these group of cichlids, anywhere from 300 to 400 species in each of these lakes. So this is considered one of the most remarkable, what's called an adaptive radiation that has been extremely recent and extremely accelerated, and there are a lot of weird fish in there, and this is one of the weirdest ones. This is a species of fish that is what's called a cichlid. They're fairly common even in Central South America and Africa. They're very common. They're extremely diverse, and they have a lot of unusual eating habits. This species is called the scale-eating cichlid. Guess what it eats? It eats the scales of other fish. So it's a predator in some sense, but it doesn't kill the prey. So what it does is whatever it's, I can't remember, there are particular species of other cichlids that it likes to come up from behind, right? And then go to one left side or right side of the flank of that prey and go and dart in and eat it, pull the scales off it, okay? And presumably, I assume it gets a little muscle with it, right? I can't imagine they can just eat. So anyway, what they're doing is they're coming up from behind a potential predator, and they can choose the left or right flank of the prey to eat from, okay? Then when you look at these things, same species, same lake, same populations, all schooling together oftentimes, it turns out one of them has what's called, the jaws aren't straight on, they're oriented to the right or left side. So one of these is called what's the dextral side, or kind of right-sided, and sinistral, the left-handed, the sinister people. You know, left-handed people in general are sinister. Right? Are you a lefty? Yeah, yep, okay. So sinistral morph, you can see its jaws, it's easier to see here, right? Its jaw is not straight on, but oriented that way. The dextral morph is oriented that way, okay? So you go in, this person studied these populations, went in there and calculated the frequency of these two phenotypes within the same species in a single lake, and guess what they found? It's pretty much around 50-50, and it turns out it kind of oscillates a little bit around 50-50. So let me show you, they did this study. This is a Japanese group, published it in Nature, and I don't know if anybody's done anything beyond this. This is what's called negative frequency dependent selection. This is one of the outcomes. Negative frequency dependent results in sort of, in this particular case, intermediate frequencies of each phenotype. So here's the frequency, we're just saying, of the sinistral individuals in this population. And they did this study from 1980, over a 10-year period. It's easy to do. You go out and catch them in a net, count them, throw them back in, right? Do this every year. Look what they found. Does it look like you're eliminating one of those phenotypes? Nope. They're kind of reaching an equilibrium in general. It's kind of oscillating around an equilibrium value about 50-50. But you can see, is it exactly on 50-50 all the time? This is what's called a stable oscillation. You will be talking a lot about these. But what you notice, it goes from, the sinistral morph is above 50% here. What happens over 3 years? It goes down a little bit. Oh, it's rarer there, right? then it goes up. Oh, it's more common, then it goes down. Oh, it's rarer, then it goes up. This is what's called an advantage to being rare and a disadvantage to being common. So when it's common, when it's above that equilibrium at 55%, there's a disadvantage. And then it declines over time. So this is what we call negative. This is a classic example of negative frequency dependent selection. The rare phenotypes are favored. The more common phenotypes are not favored and decline in frequency. So this is a classic example of negative frequency dependent selection. Now the question is, why? Yeah. So what you really want to know is, what do you think's going on when these sinistral individuals become common and you're something that's being eaten by them? So you're a sinistral individual, right? Your jaw is oriented this way. Which way, what flank side are they eating the prey? What are they attacking? They'll be eating the right side. If you tried to go oriented this way and eat off the left side, you wouldn't do a good job, right? So what happens is, when this is common, right, those prey are actually expecting, they did all these behavioral studies, so that when this thing is common, when the sinistral cichlid is common, guess what the prey start doing? Yeah, they start watching their right-hand side and avoiding attack and doing some sort of defensive behaviors to minimize their attack. Guess what that then leaves them open to? This is the theory, to attack by the other morph, okay? So the idea here is that the reason this happens, these are at a disadvantage because they now are trying to guard the flank they're more susceptible to, and then that opens them up to being eaten by the dextromorph, and then guess what happens to the dextromorph? It gets more food, more offspring, and then its frequency increases. Okay? Is that weird? Okay, so yeah, that's one of my favorite examples. I don't know, this was done a long time ago. I don't know if they followed up on it. Okay. But again, what this illustrates is all these things, this results in this not exactly stable. It doesn't go to 30% and stay there. It oscillates around it. And the idea here is there's an advantage, in this case, to being rare, a rarer phenotype, and there's a disadvantage to being a common phenotype. Okay. Okay, don't look at that. Okay, here's what I want to leave you with. We could do this now. Okay, let's do this now. Hand it in. Oops. There. Okay, there are all the models of selection that we have talked about today. Dominant, favorable, incomplete dominant, recessive, heterozygote advantage, and negative frequency dependent selection. What I want you to fill in each one of these cells and fill in the outcome. And what I want you to say there, fixation of the favored allele or maintain both. Okay? That should be pretty straightforward, right? Then what I want you to do, and this will be for comparing 1, 2, and 3, I want you to tell me the rate at which the fixation occurs. And what I want you to focus on primarily is, you can contrast it in the early stages when it originates versus as it approaches fixation. Okay? And this part, you really only have to deal with those 3. Okay? Does that sound straightforward? And you can do it in groups or individually. So just when that's done, just turn it in and the outcome should be fixation or maintaining both alleles, fixation of the favored allele or maintaining both alleles, polymorphism. Under those 5, what outcome do you get? So that's kind of the end game, the end product. So for the rate, you only want to just kind of say, you know, for just the top three, the dominant, favorable, incomplete dominant, and recessive models. Yep. Huh? Number for heterozygous advantage. Het advantage over dominance, either one.